Welcome back. So last time we were all together, we did our 4-streak method that yielded single colonies. And so today we're going to take that pure culture, a single colony, and identify it using genotypic techniques. If you remember those from class, that's utilizing DNA to identify the bacteria. And so what we're going to do today is a reaction called the polymerase chain reaction, or PCR for short. And what that's going to do is we're going to utilize a little bit of bacterial DNA along with these probes that we call primers. Now, primers are unique to the sequence of the bacteria that we're trying to identify. And so if you remember from class, we are using a set of primers called the 16S rRNA primers. Now, all microbes have ribosomes, therefore they also have their own 16S rRNA. Now, there are certain regions of the rRNA of bacteria that differ between different genuses and species of bacteria. And so what we need to do, of course, is we need to make a lot of that DNA in order to have enough to send it off to sequencing so the sequencing people can identify exactly what the bacteria is for us. And so we'll cover sequencing in another video, but we're going to take that sequencing, compare it against a database that has all different species, their genes known to man in the database, and we're going to use that PCR product that we get to actually compare it to the database that we have sequences in. Now, the polymerase chain reaction, of course, needs a couple of components. Right here we have this tube that's been pre-prepared called the master mix. What's in here are the primers, some water, some dye for running it on a gel, and as well as this enzyme called polymerase. Now, polymerase is going to make more DNA for you, and it also has all the components of DNA, like A's, T's, G's, and C's. And so that enzyme is going to copy more DNA if the primers match the actual bacteria that you have in here. So to set this up is a relatively simple procedure for a very complex set of reactions. And so you're going to take your pipetter, you're going to set it to either 20 microliters or 25 microliters, depending on what your instructor instructs you to do. And you're going to take your master mix and suck up 25 microliters. Okay? And you can see how that looks in here. Then you're going to put it in to your PCR tube. Now, this PCR tube is a very, very tiny tube, as you can see by the size of it and the size of my hand. And you're simply going to put that liquid in there and shut it. Now, you might also be wondering, well, Angelo, hold on, you did that wrong, because note that that tube's unlabeled, and you would be correct. So the first thing you do before this, of course, is label your tube. Now, it's very specific how we're going to label these, because remember you had a master plate last time, and that bacteria that we're testing today that we have single colonies from is from that master plate. So you're going to, of course, hopefully remember your naming pattern. So note that you had bacteria of 1 through 20, right? So what you're going to label on this tiny tube, on the side of the tube, is, of course, your initials, so we know whose master plate this came from, and then the number that corresponds with your bacteria, okay? So if your initials were AK like mine, I would write AK and say the bacteria number from my master plate was 2, I'd write the 2 on it. So again, we can consistently reference back to the master plate for what we actually are trying to work with, okay? And that's all. You have to have this labeled on the side. Some people prefer to label it on the top. With these types of tubes, that's going to be a very tricky thing to do because it's a domed lid, so I recommend on the side. Once you have that, now the only thing that's missing is the bacteria. So to pick up some bacteria, you'll need a new pipette tip. And all you do with this pipette tip, you don't even have to squeeze the plunger, is you locate a single colony on your plate and you simply press it, okay? Now, you don't need a whole lot. There's 100 quadrillion bacteria in that little spot. And then you take your pipette tip that has some bacteria on the end, and you simply pipette up and down. You can even swirl it around if you wish, and that is enough to get all that bacteria in there. You close the lid. Okay? So now that your bacteria is nice and mixed in there, either by moving around the pipette or pipetting up and down very gently, now we have that reaction ready to go. Now the next step is going to be interesting because we're going to use this machine right here called a PCR machine, or some people call this a thermocycler. The way this works is it is going to heat up this reaction to do 3 different sets of reactions within this tube. The first step is to heat it up to about 94 degrees. This is called the denaturing step. What that's going to do, of course, is going to separate the DNA. Remember, DNA is double-stranded, so it's going to separate the DNA into 2 strands to allow the primers that we have in here, the other pieces of DNA, to bind to the right region of the DNA. Then we're going to cool it down to a certain temperature. That temperature might be between 58 or 55 degrees. Again, you're going to check with your instructor. And what that's going to do is going to allow the DNA primers to bind to the actual strands of DNA. Now, once that happens, we have to heat this up again to about 72 degrees Celsius. That is the proper temperature of the enzyme to make more DNA. And remember, it only makes more DNA if the primer's bound to the template DNA that we have in here. Okay? And so that's going to allow us to what we call elongate the DNA pieces. Now, that's only going to do it one time, so that's going to double our amount of DNA, but we're going to do this 30 times. And so that's going to allow us to grow DNA exponentially, right? We start with 1 copy of DNA, then we go to 2, then we go to 4, then we go to 8, then we go to 16, so on and so forth. So by doing this, in about an hour or 2, we'll have 100 billion copies of DNA. And so that's going to be enough to do our next step. Now, in order to do this, I'm going to come around to the front. It's a very, very simple self-explanatory machine. You click this button that says Setup Run, or Open Method, and it will say some various things. The one that you want is called Colony PCR. So when you click Colony PCR, all the parameters are nice and set. You click the Next button, and when you're ready to load your reactions, you simply open it up. Now, very importantly, there's this black cassette in here. This is supposed to stay in here. If you were to put this simple tube right against this metal, it would melt. And so you don't want to melt it. So this should already be preset in here, so you shouldn't have to worry about it. But if you see that this is not in here, make sure you let your instructor know. Okay? And then all the class is going to load this at the same time. So please do not click the Ready button until the entire class has their samples in here. You simply put the tube in, close the lid, you're ready to go, and you're starting run. And the rest is going to be self-explanatory. And so the next thing we're going to do is, once that's all done, is going to allow us to run a gel with it. And so I'll see you in about an hour and 38 minutes.